Hello students, welcome to botany class. So today we are dealing with the first year chapters, first year topic, that is the sexual reproduction, which is very important topic in the competitive exam. So in this sexual reproduction, how many questions we can expect? As it is in each and every chapter, two questions. Apart from that, this sexual reproduction chapter is going to be combined with the taxonomy, that is the families. Okay, Fabaceae, Solanaceae, Liliaceae, those three families, and apart from that, Malvaceae and the Brassicaceae. And word problems they will ask from this. So we can expect this year the word problems. Okay, and the next one, cell division chapter is going to be linked up with this. So we have to combine the three chapters, and this has to be dealt like that only. Few topics are having the completely like that link up topics, and the other topics from this chapter are the direct questions. There is the pollinating agents and the contrivances for the cross pollination. That means how they will opt only the cross pollination when the flowers are the bisexual in nature. That is the contrivances of the cross pollination. And the types of ovules, examples, again, they can combine from any topic. That is the whatever the topics we are having, whatever the examples we learn, all those examples can be combined in this. So this is somewhat the biggest topic in the NEET wise also, we can say. That is in the EAMCET also, we will get the word problems and the same kind. So there is no difference between the EAMCET and the NEET from this chapter. So first of all, we'll go with this in this chapter, the one that is the microsporangia. So the sexual reproduction. Sexual reproduction means fusion of the male and the female gametes to form a diploid zygote is called as the sexual reproduction. So this sexual reproduction will be seen in the dicot plants, that is in the dicot and the monocot plants both. And in the angiospermic plants only we are learning here, not in the gymnosperms, not in any other plants. Okay, only in angiosperms how the sexual reproduction occurs. So in this flower, which is having the bisexual nature, that is the male and the female sex organs when they are present in one single flower, the outer wall is nothing but the calyx. So that is the sepals. What we can say is the sepal, calyx. This is the outer wall. And the second wall is the petals. That is the colorful corolla. Okay. And the third wall is the androecium. When we take the flower, then we'll see like this. Okay. In the taxonomy also we learn how many walls these parts will be present in the flower. So the outer wall is always the calyx, otherwise the sepals. And the second wall from the outside is the petals. That is the these two are the non-essential floral parts because they won't participate in the process of sexual reproduction and the fertilization or anything, but they help out in doing this process. Okay, so the essential floral parts are nothing but the androecium and the gynoecium. So those are present in the third and the fourth wall of the flower respectively. And now we are seeing about the androecium. So the androecium is having the two parts in it. That is the filament and the anther lobe. And this filament is which is attached to the thalamus at the base. This is the anther lobe. And this is the filament. Filament is the attached to the thalamus at the base and which absorbs, takes up the nourishment for the further development of the anther lobe. Inside the anther, now the microspores are going to be developed. That is the pollen grains which were released from the flower. Those are developed in the anther. So how this anther is present in different plants. So we are having two different types of plants in the entire plant kingdom. Dicots and the monocots. So in this, which type of plant is having which type of anther? That's the most important one. In the monocots, it is having only one lobe for the anther. And this lobe is again divided vertically to make the 2 theca. Okay, so in each and every theca, one microsporangium will be present. So like this, total 2 microsporangiums are present in the monocot plants, and that is the anther of the monocot plants. And whereas in the dicots, dicots are having 2 lobes. These 2 lobes are present for the dicot. Each and every lobe is having 2 theca. Na? So again, it is having the 2 theca. It is divided like this. And this is also divided like this. This is 2 theca. This is 2 theca. So total, it is having the 4 theca. Okay, when it is having the 4 theca, so if we see from the outside, from the upper side, then we can see it is having like this. Like this, 1, 2, 3, 4. So these 4 are the 4 thecas that are present. And in this, each and every theca, one microsporangium. Like this, 4 microsporangiums are present in the dicot plants. And these microsporangiums are having the capacity to produce the microspores inside them. So the microsporangium is covered with the anther wall that is made up of 4 layers. So outer the anther wall, here, labeling also they can ask us. And this microsporangium is having the 4 anther walls, 4 layers of the anther wall. The outer one is always epidermis, which is the single layered epidermis. And the second one is the endothecium, which is having the fibrous wall thickenings in it. This is the fibrous wall thickenings, which gives the mechanical support for the anther. And this, and below this fibrous wall thickening, that is endothecium, middle layers are present. These middle layers are always 2 to 7 in number. Okay, and the last wall layer is which is surrounding the microspores, otherwise the sporangium, is called as the tapetum. So the tapetum is bilayered cells. That is the not bilayered, sorry, binucleated cells. So binucleated means it is having the 2 nuclei in each and every cell. That is the characteristic feature of the tapetum. And the main nature of the function of the tapetum is nutritive function. That is, it supplies the nutrients for the further development of the microspores in the microsporangium, as well as it helps in release of the microspores from the microsporangium into the anther. So these are the 2 functions for the tapetum. And they will ask what is the layer that is attached, that is protecting externally to the microsporangium. That is the tapetum, what we can say. And the binucleated layer in the wall layer of the anther, that is also tapetum. And how many layers of the wall layer are present? More than 2. There is nothing but the middle layers. And due to the development, during the development of the microspores, one wall layer will disintegrate. Which one? That is also middle layers. So once the microsporangium develops, expands, once it is expanding, it pushes the middle layers towards the endothecium. The endothecium is having the fibrous wall thickening, so it won't move. It will be pushing the same pressure on the middle layers to move towards the tapetum. So between this movement of the middle layers, they will be crushed and becomes a powder-like structure, which helps out in the further protective nature for the microspores which are produced in the microsporangium. So how these microspores are produced in the microsporangium? In the microsporangium, microspore mother cell, which is one of the cell which is deployed in nature, will act as the microspore mother cell. This microspore mother cell, will call the microspore mother cell as the MMC. And this microspore mother cell, first of all, undergo the meiotic division. It goes with the meiotic division. This is the microspore mother cell, MMC. which undergoes meiotic division. That is the reduction division, where 4 haploid cells are formed. And these 4 haploid cells are called as the 4 spores, which are present in the tetrad condition. Initially, all these 4 are attached haploid. Okay, all these 4 are attached to one another in the tetrad condition. After certain development, those will be free, and each and every microspore now starts dividing mitotically to increase its number. This is the increase of the microspores in the microsporangium. Like that, once it expands, it pushes the tapetum towards the middle layer. Like that, the middle layers were crushed and becomes powder. After attaining maturity, the tapetum will dry off and it releases the microspores in the anther, in the theca itself. Okay, in the theca. Now, in the theca, when these are released, the powder-like structure that is present surrounding them, though that will be attached to this microspore and becomes very thick in nature and makes the outer cell wall for the outer protecting wall for the microspore. And this protecting wall will be keep on touching the stomial cells which are present in between the two theca of the microsporangium, like this. And in this one, the cell walls are very thin and the long elongated. So they can be easily released. Otherwise, they can be easily split at that point, and the microspores were released into the outside atmosphere, called as the pollen grains. Once it released outside the atmosphere, immediately the outer spore wall that is present inside this this will become the spiny in nature, rough spiny nature of the spore wall, called as the exine. And this exine is made up of sporopollenin protein, and which is very rich in the proteins and the enzymatic action. And this is highly protective in such a way that it can protect the pollen grain from the outside atmospheric changes, as well as the drought conditions, as well as the enzymes and the chemicals that are present in the outside. So everything will be present in outside, but this pollen grain cannot be infected or affected. So that is the exine nature. And below the exine is a smooth intine will be present. That is the black colored line. This is called as the intine. This intine is made up of the cellulose and the pectin, which is giving the much more protection for the nucleus that is present in the microspore. So this is nothing but the now pollen grain, what we will call. This pollen grain will be released from each and every plant in 2 different types. That is the bicelled pollen grain, otherwise 3-celled pollen grain. What is meant by bicelled? In the maximum plant, that is in the 60% of the plants, they will be releasing the pollen grains in the form of the bicelled. So how the bicelled? So this pollen grain will undergo the simple division, unequal simple division, making 2 nuclei or 2 cells which are unequal. One is the bigger one and another one is the smaller one. So this is called as the vegetative cell and this is called as the generative cell. And this is this is the 2-celled stage of the pollen grain, which is released into the outside atmosphere. In some cases, this generative cell will undergo mitotic division and makes the 2 male gametes. 2 similar sized male gametes. So that is the 3-celled condition. Whatever may be the pollen grain which is released into the dry atmosphere travels through the air and reaches the stigmatic lobes and starts its further development as the pollen tube, which passes through the style and reaches the stigma. Okay, reaches the ovary. And here, these pollen grains are having very rich in the protein content in the exine. So this protein content is going to be taken up and stored in the pollen banks. And this protein banks are the protein can be stored, and the pollen grains are also can be stored in one container, otherwise in one place. So that is called as the pollen banks. And these pollen grains were preserved at a temperature in the liquid nitrogen minus 196 degrees centigrade. At this temperature, liquid nitrogen, in that liquid nitrogen, we can store the pollen grains for a period of time, for a long period of time. That also years together, we can store them. And whenever we want to go with the artificial hybridization, we can take up these pollen grains for doing the artificial pollination and the fertilization and the hybridization process. So in the tissue culture, these are going to be utilized. So the next part will continue in the next class, right? Thank you.